Hey you guys! Today we're going to talk about the electron transport chain, which is the last step in cellular respiration. Remember a couple days ago on Monday we talked about glycolysis, and that's when glucose gets split into pyruvate. Um, After that, the pyruvate has two choices. It can go on to aerobic respiration if there's oxygen, and if it's not, it will go on to fermentation. But ultimately, you haven't really released a whole lot of energy from pyruvate at the point when glycolysis ends. So oftentimes when there's oxygen and you're a multicellular organism and you have mitochondria, you'll go on to the Krebs cycle, which we learned about last class. There is sort of a precursor step called oxidative decarboxylation, but at the end of the Krebs cycle, you've got another ATP or two and a whole bunch of NADH and FADHs. And that's when we move on to this stage, which is the electron transport chain, otherwise known as oxidative phosphorylation. And this is when we get the most ATP. We're going to hopefully get about 30 ATP in this process. So this is Act 3, and it takes place in the inner mitochondrial membrane. So this right here is a mitochondria, and remember it has the matrix, which is that intercellular space, and then it's got this folded membrane called the inner membrane, or the cristae, and embedded in this membrane over here are a bunch of proteins, and they're going to help the electron transport chain happen. And there's this space between, kind of this rosy colored space, that is the inner membrane space. But just to repeat, the NADH and the FADH2s that you made primarily in the Krebs cycle and a little bit during glycolysis are going to get oxidized. And if you recall, they're going to lose their electrons. Leo says GER. So they're going to lose their electrons, get oxidized, and release a whole bunch of hydrogen ions, which you can see right there. They're going to pump them into that inner membrane space and create a concentration gradient where ATP synthase can come back on the scene and help make some ATP. So right here is a picture of the electron transport chain. And here is NADH. You can think of NADH as a taxi cab, and it's got two guys in it who want to go to a dance, let's just say. So the taxi cab drives them to the entrance and drops them off. The two guys go in through this door, and as they go into that protein, they grab a date. And in the case of an electron, what do they like better than a hydrogen ion? So they pull the hydrogen ion into the inner membrane space, but they don't actually go, the electron doesn't actually go into that inner membrane space. It bounces to another protein and then another protein, and every time it goes through these bigger proteins, it brings another hydrogen ion in. And finally, at the end, the guys say, you know what, after all, I don't think I want to go into that dance. It's not that exciting. So they wait at the door, and oxygen comes along, aka mom, grabs them and turns them into water. So meanwhile, there's all these hydrogen ions inside the dancer in the inner membrane space. There's all these hydrogen ions, and it's all hydrogen ions. And eventually, when there's more hydrogen ions in here than there are out of here, those hydrogen ions bust to move out through ATP synthase and generate the production of ATP. Remember, there is another electron carrier involved that was made in the Krebs cycle, and that's FADH2. And FADH2 brings a couple of electrons, but those electrons enter a little bit later in the electron transport chain, so they don't bring in as many dates slash hydrogen ions, but they still do bring in a bunch of hydrogen ions. So in the end, oxygen is necessary to strip off the electrons, or the guys here that don't want to go to the dance, so that so that more electrons can keep coming through the system. If oxygen is missing, then NADHs aren't going to be able to drop off any electrons because there's going to be a backlog of electrons in this electron transport chain. And if there's a backlog, then that NADH can't move, and that means the Krebs cycle can't move, and that means that no new hydrogen ions can come in here, and that's when our body says, okay, well, I can't make energy by making this concentration gradient and letting chemiosis happen, I guess I'll just do the next best thing and do glycolysis. But ideally, when these hydrogen ions come in here, they get a high concentration and they're going to bust and move out of the ATP synthase. So let me show you an animation of that. And then I'll explain a couple more things.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
So all the names of each of these proteins is not important to memorize. Just realize that this is an electron transport chain where each protein has more reducing power, the ability to gain electrons, than the other, and that the electrons are being pulled from one protein to another and ultimately being grabbed by the final electron acceptor, which is oxygen, and turned into water. So just a little bit more about how ATP synthase works. Here's ATP synthase in action.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
So that's the electron transport chain. Your homework is to try to draw that so that you understand what's happening. Remember that this is happening in the mitochondria, in the inner mitochondrial membrane, and that's the inner mitochondrial space. Oops. Anyway, I hope you enjoyed the show. Enjoy your weekend. Happy Halloween.